The topics we will cover in this presentation are basically the rules around out-of-specification investigations, the different phases based on the MHRA and FDA guidelines, and we'll also have an extra section just on outlier tests and how many resamples you might decide to use when you do retesting. There are various useful guidelines for out-of-spec investigations. There's the famous FDA guidelines, and more recently the MHRA have produced some good presentations and methodology for that. And there's the PICs aid memoir to inspection of quality control laboratories. The FDA guidance covers all the chemistry-based testing in the pharmacopoeias and the biologicals as well, but it doesn't cover in vivo biological assays. It includes testing on APIs, excipients, other starting materials, finished drugs, anything you get from contract manufactured organizations, contract laboratories doing work on your behalf, and any of your testing results you get as part of your stability trials and validation studies and monitoring of your critical utilities. You will get out of specifications anywhere in the pharma factory where there are specifications that need to be met. It sounds obvious, but there are obvious exceptions, like during stability trials and environmental monitoring and even production monitoring, where you may have some alert limits and action limits. We'll talk a bit about those next in terms of out of expectation and out of trend, but essentially finished products, starting materials, and so on will have specifications which you can get an OOS for. Before we start, I realize that there's a lot of discussion on the internet, LinkedIn for example, about out of expectation and out of trend results, and some confusion about what they are and what OOSs are. So out of expectation may be an example where there's a batch potency adjustment. So you've got the potency of the starting material and you make an adjustment, but the final result is not what you expected. So that would be an OOE, and you might investigate what happened there. Similar process to out of spec, but nothing's out of spec yet. Another case may be like a pH adjustment. Again, you've made a pH adjustment to an injectable bulk product, and it's not where you expected it to be. There's no trend to consider, so it's not actually out of trend. And you might get them in out of environmental monitoring as well. You get so some practical examples could be with for example, stability trial, where it's following a steady trend or not trend at all, just a straight line, and then there's one up and down, a down movement, but it's still within specification. Also in production, might get production processes suddenly behaving different to what they used to be or normally do, but they're still within the alert and action limits. In these cases, production QA, QC may still pull together some investigation plan to find out what is going on. The expectation is that your process is in control and that you don't just ignore things if they're still within specification. The current guidelines split the investigation into 4 stages: 1A, B, stage 2 and stage 3. The first part is the initial investigation in the lab to make sure something obvious hasn't gone wrong, the wrong dilutions or something like that. Stage B is a much more in-depth laboratory investigation. Stage 2 starts to include production and the potential for sampling, and we'll talk about those a bit more in detail following this slide. Here is an overall top-level flowchart of the process expected to be used. So you get an unexplained result. It gets reported to the QC manager. QC manager will do the initial phase 1A assessment, looking for really basic mistakes like you did the wrong sample or knocked it over or something. If it doesn't get past that, it'll go to 1B, which is a more in-depth look at the methodology, the instrumentation, the calibration, looking at validation reports, looking for still laboratory-based issues, which but much more harder to spot. If it goes beyond that, then this is the first time it becomes a combined production and lab investigation, and it's important to understand that this phase is where you do talk about retesting the same sample or potentially new samples, but it's much more complicated, and we'll get into that later. The phase 3 is a final combined production lab assessment, and that's when you discover whether it's a real OOS and the product is defective and the batch is rejected, or not. And that then goes through the kappa system as usual, and you may have reworks and things like that to do. This is a list of the stages you would go through over the course of the whole investigation. Like I said, during the early phases, it's focused in the laboratory, and there's many steps which then lead you to looking at the batch itself and homogeneity. But the key point in this slide is to show it's a stepwise process, and you don't jump into all different aspects from the word go. As far as any employee is concerned, whether it's in production or the laboratory, the employee should be trained to not, I guess, deliberately create an OOS starting machine when you know the calibration's not right, running a run of samples when the system suitability wasn't right, or you know you made a mistake in a dilution or something. There are quite a few biological assays which are time consuming. For example, the endotoxin test, and there's a kinetic step in there where you have to use a heating clock for a certain number of minutes. It's quite easy for an analyst to make a mistake there and cook it for too long, and you end up with a invalid assay, and causes a lot of waste of time. It's not actually an out of spec, but the main point is don't proceed with assays and testing or production if you know it's not going to work out in the first place. Overall, the investigation should follow a predetermined and structured plan. I mean, that's what the SOP should allow for, and the finished product should be the report, which can lead to product release and any kappa or change controls that are required. The investigation should always look forward and look back, in that if it's an analyst specific or instrument specific, it may be more than one batch that's involved. Look for trends in common products and analysts, other trends in like changes of shifts and things like that, and what is the impact on materials inventory out in the marketplace, work in progress. Once you start digging into these things, the tentacles can go a long way. So now we get into the details. Phase 1, the obvious laboratory error. You know, all your samples and test solutions that you use for testing should be kept irregardless until your calculations have been checked and the results passed by the QC supervisor or manager. You should have enough samples, and the sample size should be enough to cover 3 times testing, and that doesn't include your normal retained samples. So simple laboratory errors, the analysts can get the workbook checked by a senior analyst, and they might find calculation errors. They might have used the wrong standard, or the standard may be expired or close to expiry and not look the same as it has done over 6 months, 5 months ago. There may have been a power failure, there may be like leaks in the piping on the HPLC and so on. So 1A is really looking for basic laboratory errors in microbiology and chemistry, you can basically discount the result on a factual basis. This slide simply shows more examples of the obvious laboratory areas you could look for in your initial investigation. So this flowchart simply is a breakout of the original one in more detail. So the phase one investigation, if you find an obvious error marked on the left in red, you still have to document the quality event in your QC system or the QA system, depending how your company is set up. And if it is a problem with the method, that people are finding workarounds to, something like that, then you might still raise a CAPA to correct that if it's a chronic issue. Examples of that I've had in the past where there's a laboratory method which involved sonicating samples and filtration steps and extraction steps, and all these steps were not really specified precisely in the method, and it was the senior analysts who knew the method from a long time ago, who knew what these, I guess, techniques were, and they had to be put into the method. So it's things like that that you might put a CAPA up for. If there's nothing obvious found, you go to the phase 2B, which is the more in-depth investigation. So if you're unfortunate enough to have to go to phase B and what's called an indeterminate error, that is, you cannot on initial investigation figure out what's the problem is, you have to go to phase B. The OOS should be raised and the QA manager should be immediately informed and the investigation commenced before any retesting or resampling is carried out. That's kind of a difference from what used to happen, is that QC were often left to their own devices to start with and retest and so on, but that's changed, and that's a major change because QA are seen as independent from QC, so QA need to agree to any kind of retesting and resampling. So during the phase 1B, there's the same, I guess, interrogation of the analyst and confirm the knowledge and training records. You've got to also make sure the sample was taken by the right person. Some companies don't have a specified person, but when you have complicated processes, it is important to make sure you get the sample that's representative from the right part of the process, and this is often figured out during the R&D process development stage. Also, you've got to look at all the chromatograms and look at any changes in relative retention times and the peaks. Are they using a new column? Have they just changed columns? Further looks at the instrumentation and calibration. Has the service guy been in recently and changed something and logs not being filled out? So it's a much more detailed investigation. And the idea is for the supervisor to be absolutely sure what kind of category the OOS is for the rest of the investigation. So this flowchart briefly explains what happens when you get to phase B. The QC manager would have to come up with a hypothesis as to what they think's going on with the results, and on the left hand side, if the QC has done all its checks, it checks all its records and everything checks out fine, there's clearly no assignable cause found. The only answer therefore, I guess, is the product. So production investigation is required and phase 2 starts. If the investigation uncovers an assignable cause in the lab, then they can invalidate the test data, repeat the analysis and record a new result and close out the investigation. And they would also have to raise a CAPA to prevent whatever happening again. This slide reiterates what I said on the previous one, but briefly mentions microbiology and not throwing samples out. And at this stage, once the QC results have been reviewed, and you're at 1B and you have got a hypothesis test to carry out, at this stage you can still only do these tests on the original sample or preparations of the original sample. It's ideal if you've got the original volumetric flask, for example, but if you've got the original bottle of powder or tablets or vials, so much the better. It's not time yet to go out in the factory resampling yet. This slide simply has a formal definitions, examples of the types of outcomes you might get in the 1B stage of the investigation. So when the investigation gets past all the lab stages, phase 2 starts, and phase 2 allows the lab to continue with hypothesis testing with approval from QA, and that's when the additional samples can occur. Additional samples can include retention samples, other batches, spiked samples for sort of recovery studies and things like that. And if there's a hypothesis involving sampling itself, things like that can be considered at this stage. On the left-hand side is the production side of the investigation. The deviation quality event is raised, and the manufacturing conduct a parallel investigation with QC, which might include investigating the sampling methods and things like that. Hopefully through that stage you do come up with an assignable cause, and a batch decision can be made. In so far as the lab's concerned, get into a loop with no assignable cause potentially when they conduct the investigation with the manufacturing one. So as mentioned on the previous slide, phase 2 involves the production investigation, and what's kind of changed is that the resampling and testing is not supposed to be allowed until production have finished their investigation, and even then it still has to be to a hypothesis testing protocol. So even though the lab might suspect sampling, they cannot really do anything until production have done their investigation. An example could be, if the hypothesis testing could be querying the analyst. It may be that usually a senior analyst does the testing on the product and they were on annual leave, and a less experienced analyst was testing it, although you can't really say that because everyone should be trained. So the choice is, for example, 2 equally experienced analysts or 1 highly experienced analyst. The problem with this is if you do find an analyst error, you're going to have to look at everything they've tested that's similar to that, looking back at other batches, which is quite a big problem to solve. So here's a bit more about what's involved in a hypothesis test. A simple way to explain it is you come up with an idea of what you think went wrong, whether it's in the lab or whether it's in production, and then you test it. You try and duplicate it or recreate those conditions. So even though production are meant to go off and do their own investigation, obviously there are times when production and the lab, I guess, come up with the same idea, the same hypothesis, and it might involve the sampling or where the sample was taken, and so you both need to sit together and come up with a plan sometimes. So yeah, so come up with what you think the root cause is, decide ahead of time what samples you're going to test them and why. You need a testing plan. It needs to be approved by QA, especially the order of doing things. Don't forget, any data you generate you have to keep, and you can't just throw results away if you don't like them. So retesting usually in phase 1 and 1B requires using the original sample supplied by a lab, or insufficient remain sample, go back to the same production container without further mixing. If you get to phase 2 and you think there's still sampling issues, you still should only be going to sample from the original part of the batch. Production may come up with some other ideas or investigation issues. There may have been a mistake in that case as well. So the resampling plan um, could be informed by production, and they may know for sure that, for example, with tablet coding, that a nozzle was getting blocked between 7 and 10 and with multiple like stoppages. Even though there should be SOPs for this, sometimes things can get through without being noticed, I guess. So there's no there is no ultimate plan. Every situation will be different. So there is no template for a perfect hypothesis test. It depends on your process. So here are some more examples of the ideas or hypotheses that might come up. Checking equipment, checking sample preparation, checking the sampling. The main point is you need to find a probable cause and prove it. You can't exclude the original result unless there's a clear laboratory or production sampling error revealed in stage 2. That's the end of the story there. So here's some additional information on the sample. Um, I've talked about it already, but the key point is before you start getting new samples and resampling, you really must prove that the original sample has got something wrong with it, or if you duplicate the same results, then the sample's correct, or your method. So it's not just a case of sampling and testing until it comes back into spec, as a lot of companies used to do. And you need to justify the number of retests as well. The famous bar decision in the US came to the conclusion that the minimum number of retests you need between 5, 7, 9 for it to be statistically significant. And you can't discard the original result unless you have a reason to. It may be part of that same sample set, but on the limits of the standard distribution. So here's an overview of the final process. So we've talked about phase 2, the hypothesis protocol. Taking the left-hand side, for example, the lab confirms the out of spec. Basically, the investigation stops there, and QA have to decide what they're going to do with the batch. In the center route, there's no assignable cause. They've done retesting, resamples, averaging, considered outliers for biological assays only. Report the investigation. Again, batch decision. This is a more tricky one where there's no assignable cause. There's no real short answer to that one, and QA and production and R&D need to discuss what is possibly going on there. The right hand side, another good scenario is there is an assignable cause. The results and report are written. They're all reported, and QA decides on the fate of the batch, whether it was in spec, out of spec, or how you're going to treat the results. Would obviously result in raising a kappa as well. A lot of the problems with out of spec investigations, and the ones that auditors regularly pick up on, is inappropriate averaging of results. So assuming you've gone through phase 1A, B, 2, and you now have a set of numbers, what do you do with the results? Averaging is normal with HPLC results and microbiological assays, and for example well-based LAL testing. And it's valid so long as unless there's clear variation between the replicates of the same sample, because this means there's actually some homogeneity kinds of issues. You can't use averaging where it's to detect homogeneity, like uniformity of content and in-process blend testing, because that's the whole point of the assay. So the final result of this is regulatory authorities and auditors really do not like you to averaging out of spec results with in spec results and dragging the average in. That is clearly basically testing into specification, or using inappropriate statistics to bring something into specification. Statistics is often used. You can use a student's t-test to show the variability of the averaging, and outlier testing, but mainly for micro and biological assays. We'll talk about outlier testing later, and I'll explain why it's less appropriate to use them in chemical testing in the lab in pharmaceuticals. There are some expectations around how long all this should take. So usually the 1A stage initial lab investigation is less than 1 to 2 days. The more in-depth lab investigation may take 2 to 7 days, but it's rare that a factory production QA will not want to know about what's going on here by that time. The second stage, 1B and 2, could be anything like 2 to 3 weeks, but there is an expectation by the FDA, for example, that the whole thing should be figured out within 3 working weeks. So, when we get to phase 3, depending if the results in spec, out of spec, might not actually make any difference. You still might have to have this investigation package. R&D subject matter experts should all get together to review what happened if it gets to phase 3. Depending on the outcome, production may need to be suspended if it hasn't been done so already. You don't need to have an OS to suspend production if you think there's something going wrong. Once the batch has been rejected, there's no boundary as to how much testing and experiments you can carry out to see what happened. You cannot resurrect the batch as a result of this particular testing. The impact of the OS should be extended to a thorough investigation of other lots that have gone before it, that are in process, that are in the market, may have been sent to contract packers, contract manufacturers, and so on. Then you can also, depending on your procedures and the type of products you make, you may be considering rework procedures, or for example in the case of the tablets where there's been spraying issues with the coating, you might actually be able to salvage some of the batch by segregation and resampling, things to that effect. If no lab errors are identified and the OS stands, all results should be reported and all data used in the final decision. If investigation finds that there is an inherent sampling error and a new method is needed, then a new method can be approved via change control, and again a look back at process validation, risk assessments, etc. So as you might gather now, the initial OS doesn't mean the batch fails. There's various phases of investigation to go through, and the OS ultimately needs to be proven to affect the batch quality, safety, identity, purity, identity, potency, and meet the specification. It still doesn't mean it needs to be rejected. I mean, this procedure applies to APIs as well, for example, and there's quite a lot of opportunities for reprocessing in APIs. But ultimately, the decision is QA's, and it's the decision's got to be made with all the information to hand. In some rare cases of drug shortages, QA can discuss with the regulatory authorities the situation, and if it's a very borderline technical, rounding issue, fail and still within expiry, there may be some leeway with the regulatory authorities, but that's very rare. So at the end of the day, when the investigation has been completed, you're expected to have full and complete, accurate records, passing data integrity regulations and everything for the whole OS process, and this will follow through hopefully to deviations and CAPAs, etc. Make sure the whole quality system loop is closed, and you have your investigations, your root cause analysis, risk analysis, and the final disposition. That way, when you present the case to an auditor, it is not disparate bits of paper in the lab and micro and QA and production. Is all actually already compiled in a full and documented report, and it has a scientific rationale. If it's presented well, the auditors will have a favorable view of the system, but if it's all over the place, they will have their alarm bells going already. Subject that generates a lot of interest is outlier testing. People generally do not suggest that you can use this for chemical testing. Some people think it's actually the rules, and certainly in pharmaceutical contexts, I tend to agree. But historically, it actually was invented by chemists. In non-pharma industries, it's quite normal to be dealing with hundreds, if not thousands, of samples. For example, in the oil industry and the mining industry, an example I'd give is a company prospecting for gold will take thousands of coarse samples from the soil, and they would assay the soil samples, and you would get a standard distribution of the concentration of gold ore or iron ore in the soil. Occasionally, though, you might have a sample which is pure gold, a gold ingot. That doesn't mean the whole mountain is made out of gold. It means you found a pocket of solid gold. So it's a different sample population from a statistical point of view. So in the initial test, that would be an outlier, but then you would separate it into a different subgroup of gold, solid gold. So the point I'm making there is, just because you get an outlier doesn't mean you want to reject the result. It's giving you information, and this is the whole philosophy behind outliers. Even in the pharmaceutical industry, is it's a good test to do to see if there's something very, very strange going on, or whether the sample is from a different sample population. But it's not really of any use for rejecting results, so that you can pass a batch, which is where it's misused. So for the pharmaceutical example, because you're expected to have thoroughly developed the product and have an operating envelope and develop critical process parameters and critical quality attributes, the idea that you'd get outliers is kind of causing problems for your validation. So here are the extracts from the original text on outlier testing, and just formally saying that if you've got a small sample set, then having an outlier is quite a delusion, because it relies on a standard distribution, and the pharmaceutical validation supports a standard distribution, otherwise the product wouldn't get validated. So this is kind of the loop you get into. The USP does allow it for biological assays, because biological assays for antibiotics and similar tests are well known to be variable, and biological assays behave in a manner all of their own. This slide just reiterates, in the pharmaceutical industry, you really can use outliers for chemical tests using primary or secondary standards, because they're based on chemistry. There's no biological variation in there, and ultimately an outlier test cannot identify the cause of an extreme observation and cannot be therefore used to invalidate a suspect result. There are several outlier tests. There's Dixon's one, and there's one called the Q test for sample sizes below 10. It's actually quite simple. You have your observations, and you put them in ascending order, and the outlier test is basically the difference between the farthest result, the one that you think is the outlier, and its next nearest result, whether it's at the high end of the group of numbers or the low end of the group of numbers. And you basically work out what's called the experimental Q value, and it's the ratio between the difference between the highest and the lowest and the range. In this example of the Q test, there are 5 results in ascending order. It's queried whether 56.23 is an outlier. The difference between 56.23 and the next nearest is 0.15%, and the range over the whole set of numbers is 0.28. The Q value works out at 0.54. So for 5 samples looking on the table, the 95% confidence is 0.73, which is greater than 0.54. So it's not classed as an outlier in this case. The R test, or the Dixon's test, is used more often, and it can be used for different sample sizes, so the table varies slightly as the sample size increases. So using the Dixon's test, you can see here for different sample sizes the equation changes slightly depending on what range or what the difference is you're trying to work out, but the use of the table is exactly the same. In this example, using the R test, again you find for the sample size of 4, the 95% confidence, the critical value is 0.765, which is greater than the calculated value of 0.55, so the result is again not an outlier. So here are some examples of the kind of citations you get for OSs, and if you read them you'll note that a few of them actually relate to not using your procedure properly. So the last one, releasing after OS using a grand average, including in spec and out of spec results. You can't average them. The prior one, not reviewing all the batch records properly the way you're supposed to. So it's not just having a out of spec SOP, it's making sure you follow it in exactly the sequence that you say you are. And to finish off OOS, here are some of the findings and judgments of the original bar decision that happened in the mid-90s in America, and it started the whole subject off really because the company was found to be testing into compliance, averaging bad and good results, sampling and resampling, testing and retesting, no end in sight to how much sampling and testing you can do. The judgment was that you're not permitted to average out of spec results to get a pass. You're not permitted to conduct multiple retests without any predetermined limit. So you need to say if you're going to do 3, 5, 9 repeats. And also on the outlier testing for chemical testing, you can't use it for like chemical assays. You can use it in biological or biotech assays, but not in chemistry, because your product is meant to be homogeneous and representative. So thank you for listening to this presentation, and if you have any questions regarding out of spec investigations or outlier testing, or indeed any other GMP GLP questions, just send me an email. I can also provide SOPs and forms and templates if you like. Hope you enjoyed it. Thank you.